Good morning, one and all. We welcome everyone to the first day of the two-day international conference on the topic recent trends and hurdles in physical science research, organized by the Department of Physics and IQAC, Chrysler College, in association with KSSTM. Scientific research can reduce superstition by encouraging people to think and view things in terms of cause and effect. The world is changing rapidly, so are the research areas. New and innovative synthesis techniques, sophisticated methods, and above all, huge computational power all has contributed to bring research to new heights. The renowned speakers will disseminate innovations, trends, and hurdles in this field. Before commencement of the session, let us invoke the blessings of God with a prayer song.
Make me a channel of your peace. Where there is scepter, let me bring your life. Where there is darkness, only life. Where there is sorrow, ever joy. Oh, Master Grandeur, tell me never see. So much to be consoled and to console. To be understood as you understand. To be loved as to love with all my soul.
Let us start the conference by inviting our beloved HOD, Dr. Benny George, who is the guiding light in all our endeavors to deliver the welcome speech.
Good morning, everyone. As Arundhati Roy mentioned in one of her articles, pandemics have always forced humans to break with the past and imagine their world anew. This one is no different. It is a portal, a gateway between one world and the next. We have chosen to walk through it, creating a new normal. Everything is witnessing a drastic change. Science is not one dimensional. Many contribute in many different ways, and it is difficult to say who contributed more. I feel really proud and delighted to welcome you all to the two-day international conference on recent trends and hurdles in physical sciences research, organized by the Department of Physics in association with IQAC, Chrysler College. Kerala Science and Technology Museum was established to improve the scientific temper in the minds of the young. It has a legacy, and I feel extremely happy to welcome the person who is maintaining that legacy with utmost care, Dr. G.P. Padmakumar, the Director of Kerala Science and Technology Museum and Planetarium. He has held many prestigious positions. We feel privileged to have here today for the inauguration, sir. On behalf of Christnagar College family, I welcome you, sir.
Thank you, sir.
Next, I would like to welcome Dr. Jinu Rajan. It was his unwavering support as a member of advisory committee that made every difficult task a piece of cake for us. He is presently a senior lecturer in Cardiff Metropolitan University, UK. Welcome, sir. I offer my heartfelt welcome to my classmate, Dr. George Verghese, Research Director, Marivanis College, Trivandrum. We are indebted to you for accepting our proposal to be the reviewer of the conference. Welcome, sir. My wholehearted welcome to the two towers of strength we always rely on, our director and manager, Father Dr. Tito Verghese, CMI, and our principal, Dr. Jolie Jacob. It is your determination and devotion that makes everything a grand success here. Welcome, Father. Welcome, Mom. I also heartily welcome our beloved Bursar, Reverend Father Subin Kotur, CMI, to this conference. I would like to welcome all the allied minds who are here to join us for this conference. Welcome, dear participants. I feel happy to welcome all my colleagues and students whose diligent efforts speak today volumes on us. Welcome. Once again, I welcome you all to this conference. Thank you.
Thank you, sir. I feel immense pleasure in introducing our honorable chief guest, Dr. G.P. Padmakumar, who took his PhD in environmental geotechnics from the most sought after IIT, Mumbai. He began his glorious career as a civil engineer in IHRD and then went to have an enticing teaching career as lecturer, assistant professor, associate professor, and professor at various renowned colleges in Kerala, like College of Engineering, Trivandrum. He worked as a training officer at Directorate of Technical Education. He was the first registrar of Kerala Technological University at the time of its inception in 2014. He is currently the director of Kerala Science and Technology Museum and Priyadarshini Planetarium, an institution that stands an epitome of the sense of inquiry we foster. With much happiness, I welcome you, sir, to inaugurate this two-day conference.
A very good morning to all and all. A respected director and manager of Christ Nagar College, Father Dr. Tito Joseph. A respected principal of Christ Nagar College, Dr. Jolie Jacob, Madam. Respected HOD of the Physics Department, Dr. Benny George, sir. The Research Director of Marivanis College, Dr. George Verghese, sir. Senior Faculty of Cardiff Metropolitan University, Dr. Jinu Rajan. Members of the Faculty of Department of Physics of Christ Nagar College, Dr. Sodhi P.S., Dr. Veena Gopal, Dr. Anita V.S., other members of the faculty of various departments, staff, distinguished speakers of the conference, participants, and dear students. I'm very happy and indeed honored to be here for the inaugural session of the two-day international conference on recent trends and hurdles in physical science research organized by the Department of Physics of Christ Nagar College. At the outset, I would like to express sincere thanks to the organizers of the conference and the authorities of the institution for giving me an opportunity to join with you. As the director of the KSSTM, it is my second time association with this institution. On 20th March of this year, CNC had organized a workshop on astrophysics named Cosmos 2021 in association with KSSTM. But unfortunately, I couldn't attend in person due to some unexpected urgent official engagement. But at that time itself, we have planned some joint venture programs in relevant areas. So as Professor Benny George sir already told, KSSTM is an institution which was established in the year 1984 to disseminate the scientific knowledge to the youngsters of the state, especially the students. The student means those students who are interested in scientific principles. So in order to disseminate that, KSSTM has many programs. Initially, it was a research center of Kerala University. But due to passing of time, it has got the status, but now we are again going in track to have a research center at KSSTM. To few words about KSSTM. KSSTM has a major principle or major equipment is the planetarium. So it is one of the best planetariums in India. So through that, we can have some idea about the celestial bodies, astrophysics, astronomy, etc. And also it has certain scientific galleries where the youngsters can experience the scientific principles through entertainment. And also, it has got a scientific center known as Innovation Hub in association with the Department of Culture of Government of India. So in that Innovation Hub, we have got certain facilities to do research and to promote research. So right now, we are planning to associate with the educational institutions in Kerala, which has got especially the physics and science department, so that we can have a joint ventures in the field of astronomy as well as astrophysics. I know that earlier, Marivanius College was associating with KSSTM in many things for sky observation, astronomy, astrophysics, etc. As we all know that for the last 1.5 years, the world is almost standstill due to the pandemic. As in all walks of life, it has affected the academics and research activities, except the scenario concerned with the prevention and restriction of against COVID-19. But of course, it has given a good indication for the field of research. The world was fighting with one mind to find a solution for the existence of mankind. The researchers all over the world worked together and developed methodologies and medicines within months. This has proved the firmest, showing that necessity is the mother of invention. This is the purpose with which the scientific community should approach the research activities. The research cannot be done in a room or in an institution. It should have a broad ambit. So in order to have good research and to develop good research minds, joining hands with other departments, institutions, industry, etc. are essential. So with that only, KSSTM is joining with CNC for this conference. The research should be for addressing the problems that are faced by the society, for finding solutions to the betterment of mankind. I'm sure that this conference will provide a stage for the young minds to understand what is happening in the world concerned with your branch of study, and the researchers should share their thoughts and of course showcase what the researchers have done in their region. From the theme, it is understood that the conference is covering a broad aspect of the physical science for the various materials of interest, its characterization, material structure and properties, devices and applications, simulation and modeling techniques, and of course, space science. To the students, young students, I have to point out that motivation is required for everything and especially for the research. So if you see the world scenario, Kerala is excelling in academics. We have good higher education institutions. We have good, excellent college campuses, universities, etc. But what we are lacking is we have behind the other states of India in the field of research. We have to critically analyze why this is happening. In the world ranking as well as in the national ranking, the educational institutions of Kerala are behind only due to the lack of research papers and research outcomes. I feel that there's a most difference because we have certain national institutes such as IITs, IISc, and IISc, etc. In that national institutes, we have good researchers, good research students, good research outcomes, etc. But our state institutions are well behind from that. Why that is happening? So from the +1, +2 level or from the degree level itself, we have to see that academics and research are one. We cannot bifurcate the research from the academics. Unfortunately, what is happening in Kerala is that we are seeing research is different from academics. So we are seeing academics as a way for studying something and to get marks and to pass the course or to get a degree. So this is not a way. If we can see the research as a part of academics and a part of curriculum, then we can also achieve. And also the students as well as the faculty of Kerala, they're quite inhibited to produce or present their small things to the world scenario. But if you see the other states or other countries, they will, if it is a small thing, then also they will showcase it well and they will start from that to new research. So we have to rethink in the methodology of teaching, and we have to introduce certain novel techniques to cultivate the curiosity in the young minds and let them nurture in their minds to develop a habit of questioning. So in our classrooms, the students are just listening and they are not questioning the teachers and they are not nurturing the things in their minds. So today's conference also, so many people and the researchers, professors are giving presentations and they're presenting their papers. The students should, after each session, the students should possess an ability to ask at least 5 questions to the presenters, or they have to develop, at least they have to develop 5 questions in their mind. Then they can slowly attain the level of research. So if you observe a child at the age of 3 to 6 or 3 to 7 years, at that time they have a tendency to ask questions for everything that they touch, they feel. They will ask so many questions to the elders. So that is why they are studying. So the students, especially in the degree level students, they have to develop a culture like that. So I wish all best to the to this conference, and I think that this would be a very good platform for the students to know the researchers and what is happening in their respective field of study. With these few words, I wish all success in the future endeavors of this department, this institution, and also I look forward to associate with you in future also with KSSTM. And with your permission and consent, I declare that the conference is officially inaugurated. Thank you. One and all.
It was a thought-inspiring speech. Thank you very much, sir. Now I request Dr. George Verghese, Research Director, Mar Ivanovos College, to felicitate this conference.
Good morning to all. Respected manager of this institution, Father Tito, respected principal, Dr. Jolly, my colleague and classmate, Dr. Benny, the head of the department of this institution, our chief guest, Dr. Padma Kumar, Dr. Veena, who is the coordinator of this program, and my dear researchers who are watching from India as well as abroad. I wish you all a very warm good morning. This international conference on recent trends in hurdles and physical sciences research is in fact a unique conference because the organizers has decided to address not only research problems, but also the hurdles which are facing in research, especially in physical sciences research. There is in fact conventional differences between teaching, research, and administration. A university researcher or teacher is supposed to do all these three things during his tenure. Of course, there are institutions, very rarely in India, that permits the faculties to do research alone. Albert Einstein always preferred such institutions. So that is why finally he landed at the Institute of Academic Sciences, which is supposed as a heaven for research, science research. But in our country, we don't have any such institutions entirely devoted for research. And all these faculties members are enjoying research only. Now, when we do research, we have to face a multiple of challenges, or we have to play a role of a multiple of roles as a researcher. That include doing research, publishing results, collaborating with peer teams, managing research projects, guiding or supervising scholars, evaluating or assessing the research output done by others, etc., etc. In all these, there are many issues that has to be addressed. Doing research is not a simple task. It requires thorough knowledge of the subject or in that particular field, or at least in the peripheral areas. Patience and skills are required to identify a problem connected with research, so that then only you can proceed with that problem. A researcher alone cannot accomplish all these tasks properly. The support of external agencies in the form of infrastructure, finance, resources etc. are required for doing research. Developing or underdeveloping countries like India, research happens mainly in government institutions or under government subsidized programs. Roughly 60% or sometimes more than that of the research in India is taking place in government agencies or government funded institutions. Whereas in USA, 80% of the research is conducted in private sector or with private sponsorship. So it is very difficult for countries like India to manage the support of the government because government is under heavy constraint on finance, especially when we are facing pandemic-like situations. So day by day, the government is withdrawing its support from, in fact, let me say, fundamental research. Fundamental research is the main research that is happening in universities as well as other institutions in India. Whereas applied research is happening in institutions like Space Research Organization, Atomic Energy institutions, biotechnology institutions, etc., etc. There are applied research, of course. Then facing another problem another problem that we face is publishing in the research output. Publishing or publishing mark is a great rule for any performer or any organization in research. A research without publication is in fact a farce exercise. Now people got realized about this and try to get their publications done in reputed journals as well as in books. Publications are in fact required for career advancement. It is not just for career advancement. If you publish your research, of course, others would also get benefit out of it. Whereas if you do not publish your research, keep it as a secret, then the result is that the community will not get the benefit out of it. Of course, you are using the money provided by the exchequer for doing research. Therefore, you should publish your research. Let others come and comment on your research, others come and enjoy your research output, and let others should criticize your research. So publishing is in fact very much important for the research. So where we do publishing or where we find a proper journal for publishing, that is in fact very important thing. So we should publish in reputed journals. That means reputed in the sense that journals that are being read by many people all over the globe. So then only your research work will get spread in a short span of time. Otherwise, you keep it underneath for a long time. So publishing is in fact a very important problem. So to get a publication in a reputed journal or in a highly quoted journal, it's very difficult because unless your research has some significance or some importance, the editor of the journal will not take or will not look into your publication. So we should gear up our system so that we should get a proper research output out of the research. So it is very important. The quality of the research is very important in that sense. Another important problem is collaborating with the other groups. Collaborating is good for experimental research as well as for theoretical research. Especially in the case of experimental research, collaborating with other institutions and other firms is very much important because of the finance involved. Maybe your institution is not able to support all the support for all the equipments that are needed for your research. So you can get the assistance of other institutions for conducting your research properly. So collaborating is very much needed for an experimental group for doing research. Collaborating with not only in the same institution, but also with the other institutions in the same country as well as abroad is very important. For that, you need a mobility. The researcher should move from your institutions to outside and get in touch with people outside and and expose your research to the others so that the others got interested. Maybe others got interested after reading your research out. So that is very important. Collaboration is very important. But unfortunately, while during my long career as a researcher, I have seen that collaborations are very much less in institutions, especially in Kerala. So we should promote collaborating research with other institutions. Then research assessment is very important. Who will assess the quality of your research? Of course, the publication is a great mark for assessing your research. Your peers are the best who can assess your research. Your peer groups, the similar groups that are working in similar areas are the qualified persons for assessing research. But unfortunately, for career advancement and other things, your research is being assessed by some other groups who do not know what is the actual quality of the research or actual problem that you have worked out, etc., etc. So assessing with the same team, assessing with the qualified team is very, very, very important aspect in managing research or in doing research. All these are very important issues. I hope that the conference will address all such issues during the two-day deliberations. This conference is also addressing research associated with the physical sciences. Physical science, especially a few areas have been identified. We know that every day our world is changing. Every day our world is changing due to new inventions, due to the new inventions of materials as well as its qualities. So, you know that yesterday what we have seen in connectivity has been improved. Yesterday what we have seen in computers has been improved. Yesterday what we have seen in medicines for treatment of different diseases has been improved today. What we have seen yesterday in the production of energy, especially in solar energy conversion, etc., has been improved. Why? Because of the invention of, or because of the emerging of new materials. The material science is therefore very important. So all this, I have seen that a lot of topics related to the developments of material science is included in this today conference. That is in fact a welcome suggestion. So I appreciate the organizers for taking up all these challenges together in a short two-day program. So I wish that fruitful deliberations will come, and definitely it will improve our understanding about the hurdles of research as well as our understanding about physical sciences, especially in material sciences that are supposed to change our world. So with these few words, let me give my wholehearted happiness, express my happiness as well as my appreciation to the organizers. Also, I thank the organizers for inviting me to play a small part, even though very small, but I consider it as a privilege in this conference. Thank you very much. Thank you.
Thank you, sir. Now let us listen to the recorded felicitation by Dr. Jinu Rajan, Senior Lecturer, Cardiff Metropolitan University, UK. He is the person who extended all his support in making this event fruitful.
Morning, everyone. Let me introduce myself first. I am Jinu Rajan, an academic at the Cardiff Metropolitan University in the United Kingdom. I'm glad to be the part of the organizing advisory committee of the International Conference on Recent Trends and Hurdles in Physical Sciences Research at the Christnagar College. Thank you to the coordinator, Dr. Veena Gopal, for her invitation to take part in this. The conference aims to provide a platform for young researchers and especially students to know some of the latest research topics in physical sciences. So it's a very good opportunity for the undergrad and master's students in physical sciences to know more about the research pathways in the field. And I think it is also for an opportunity for other researchers to refresh their knowledge and to know some of the latest trends in this field. And we have some very well reputed researchers in this field as our speakers, who all are kindly agreed to share their research and thoughts with all of us. And to thank you to all of them. And I also congratulate the Department of Physics at Christnagar College and the organizing committee and the coordinator, Veena Gopal, in putting together this exciting program. I assume all of you will enjoy the talks and the program, and wish you 2 wonderful days of research collaboration and engagement. Thank you.
Thank you, sir. I would like to invite Dr. Swati Miss to deliver the vote of thanks.
Good morning, everyone. Through the eyes of gratitude, everything is a miracle. It leaves smiling faces. I feel greatly elated that I was given the opportunity to express a lot of thanks on this occasion. First, I would like to thank Dr. G.P. Padmakumar, who was ready to share his valuable time with us, regardless of his busy schedule and the torments of a highly responsible position. Thank you, sir. We need a person to give us a guidance and other help whenever we feel disheartened. I thank Dr. Jinu Rajan for being a constant support system for us. Thank you, sir. Dr. George Varghese readily agreed when we made a proposal to be the reviewer of this conference. His enthusiasm was really contagious. Thank you, sir. Next, I would like to thank the pupil we always look up to with much admiration and respect. Our dear director and manager, Reverend Father Dr. Tito Varghese CMI, our principal, Dr. Jolly Jacob, and Bursar, Reverend Father Subin Kotur CMI. Their relentless efforts go a long way in making our institution the most auspicious place for learning and for being so supportive whenever we approach them with ideas and programs. Thank you, Father. Thank you, ma'am. Dr. Benny George, head of the department, Physics, has never left a page unturned because he wants everything to be so organized. We thank you for your guidance and supervision, sir. Thank you. Next, I would like to thank my colleagues and IQC team for being so devoted in everything you do. Thank you. I am compelled to use the phrase last but not the least. Dear participants, we thank you for joining us and playing an active role in this conference. We look forward to seeing you in the sessions to come with that energy of yours. Thank you all.
Thank you, Swati ma'am. Now we move on to the next session. Next, we have Dr. Chitra R. Naik, Associate Professor, Physics College of Arts and Science at Tuskegee University, Alabama. Dr. Naik completed her PhD in the area of nonlinear dynamics from Cochin University, India. She worked as postdoctoral fellow in the interdisciplinary field of bacterial biophysics and immunology at Dalhousie University and University of Toronto, Canada. She joined as an assistant professor at Tuskegee University in 2014. Her current area of research work includes nonlinear analysis of biosignals and fluid dynamics. Dr. Naik is also involved in education research at Tuskegee University. She has published her work in well-reputed international journals and is cited more than 50 times by independent authors and has a total of 148 citations and 7 H indexes. I now hand over the session to Dr. Chitra R. Naik, and along with it, a humble request to share a few experience about your above said achievement.
Thank you. So first of all, I would like to thank everyone for giving this opportunity.
It's a real pleasure. Actually, I was remembering old days at KISAT, and it's first time after a long time I'm hearing my name said right. So that gives me another pleasure. So yes, it was great. Like whatever Dr. Padma Kumar sir and Dr. George Verghese sir was talking about, I want to just hold on to it regarding the students. I feel like majority of the participants are students. So remember, one thing that Dr. George Verghese just mentioned kind of caught my attention, that we don't ask questions. Our students don't ask questions. Even when I was a postdoc in Toronto, I used to have this real fear even when I have good questions, I will never ask it because I was, it's kind of a training. So I'm hoping that but then I start to teach in Tuskegee. Here students are very different. They will write stuff in the middle of while you are doing something, and if they have to ask something they will ask it, and then it becomes an engaged discussion. So for a while I am used to it, so I was hoping that I would make some of you ask questions to me, and maybe I will number how many questions come. Okay, so one thing I have noticed recently, because I try to attend what is happening in Kerala, like I see many times there are conferences, seminars, workshops on cosmology. So initially I thought I will do something with fluid dynamics, but then I changed my mind and I decided to present a work that I'm currently doing in immunology. So how is that related to physical sciences, right? So let me start by sharing my screen. Remember, physical sciences includes chemistry. So what I thought was I want to give the students a kind of feel about how can you get into advanced research, but at the same time more relevant to the current situations. And in the end, the last slide, I have added a new topic that I have started to work on, which is health disparities, which means, disparities in racial or socioeconomic costs of, having health access. And because of that, dying early in their age. So I'm into that research recently. And how do physics come into play in these kind of situations? So that will be my focus of today's talk. So again, I am from Tuskegee University in the far south of United States. This is a historic university in the sense that Booker T. Washington, after the slavery was abolished in United States, this was the first HBCU that was set up. So this is a historic place. So, and the talk is, this is actually in this talk, I will kind of explain the hurdles in the sense like the traps that a physicist can come into because of our kind of training, and how can we apply some of the modeling that we know in explaining some of the very important topic in medical research. Okay, so differential signaling through a shared pathway. So if these kind of things cannot make us surprise, like for example, when I start my postdoc and I started looking in bacterial cell, I got so interested because till then I considered myself as a theoretical physicist and computational physicist. My work was in nonlinear dynamics and chaos. But once I started working in bacterial cell, I kind of started understanding how physics play a role in even a cell division of a bacteria. We might have heard about mitosis, like the division of the cell and all, but the basic idea they use there is actually there is an inside process where there are some proteins which move across the bacterial cell from the tip to tip, and then there is a third, so they kind of form a standing wave. And then a third protein will come and form a circle at the maxima of the wave. So then I realized there are some intrinsic processes that we can learn. And because I always kept saying I did not like biology, because I thought it was a field that is supposed to be memorized, I kind of lost on something. Of course, it's hard to pick up everything. So then I started working on thinking about or taking these aspects as more from a chemistry or from a molecular point of view, and then applying what we know, right? So that's how my talk will proceed. So the first few, I have to set up, when I am trying to tell a story, I have to set up the scene. So the first couple slides, bear with me, will be really like a theory class, okay? Like a biology lecture, the first 4 sections. But please understand that this is to make you understand what we have in a simple cell that we see. So sometimes we are so astonished by looking into space, we forget to look at what is so near us, okay? So that is what I'm trying to present today. So again, we know that you all know that especially at the time of COVID, we kind of started to understand how important immune system is, right? So we have already an innate immune system, and there are some toll-like receptors, which means, they will be always waiting, and any kind of pathogens come, they will alert the system. And once the system is, once they are alerted, what happens is there are some intrinsic ligands that is being produced, which activates the process, okay? So I will explain what it is in the next slide. So what basically I am going to look at is one type of molecule called interferons. Okay, I'm just going to call it molecules. A biologist, or if there is anyone from medical profession, they will kind of say, don't do that. But I'm just going to use it that way so that we are not confused by many terms, because too many terms in maybe a 30-minute session will not be good. So interferons are some molecules that is produced by our body to resist viral activity. So for example, when a virus comes, or anti-proliferative, that means it will prevent the division of cells, okay? So how are those common? Viral and cancer, right? It seems like, so if you have ever learned your little bit of biology, I know that I knew before. So virus actually use the our cells to divide. They need our DNA. So that means if our cell division is stopped, virus cannot replicate. So most of the antiviral treatment is to kind of reduce cell divisions for a while. And cancer, as you know, is the uncontrolled growth of cells, right? So both of them, they used to use interferons. Now, just like Dr. George Verghese said earlier, now there is a lot of new things. People don't use interferons in treatments anymore much, because it takes a toll on the body, which I will explain soon, okay? But still, there are some time that you have to use interferon. And the work that I kind of did was actually with some doctors, but again, I am looking at only the molecular aspects, okay? So what is the main question here? Again, remember, students, it's always a good practice. You see, now you have the luxury of using the chat message, and I have the habit of checking the chat even while I'm speaking, because I encourage in-class discussions. So you are always welcome to stop me, ask me any questions in between, okay, regarding this. So if you anytime you need some explanation, just stop me, okay? So why is this an important, or why is this an exciting field? So let me try to bring some excitement on you. So there are many types of interferons, okay? So why are they called interferons if there are many types? Because they share a common signaling pathway, which means they all walk the same step. But the interesting thing is they all walk the same path, but the result is different. The destination is different. So that makes it very interesting, at least to me. So again, just the name came because it's just I wondered if anyone wants to remember. It interferes with cell division, so it's called interferons. And yes, it is a primary defense mechanism. So what does primary defense mechanism means? My body itself produces it. Okay, so all our body produces interferons anyway, but sometimes we will have to give an extra dose. So there are many known type of alpha and one beta. So this is what we are going to discuss. As I said, first 3-4 slides, I think, will be like this. Now, as I said, when interferon, or when you give, like, let us say you are giving a shot of interferon, what happens is it will come and bind to a receptor. You remember, like, how does this happen? There are some cell surface receptor. There will be some signaling particle coming and attaching to the cell surface receptor. and the receptor activates. So you see, this is the molecular dynamics that I'm going to study as we move on. Okay, that is where that physics come. So some ligand or some activator, you can use that term instead of ligand. Activator comes and kind of attaches to the receptor, and then there is a downstream signaling. Okay, and it is very funny, like how these things have evolved. I don't use the word evolved anymore. Now I'm very careful because it's hard to even think about things evolving that way. So once this receptor complex is formed, there are multiple signaling pathways that can start from the same stream. Out of that, I just studied one called JAK-STAT signaling pathway, and that itself, humbles you. So again, STAT means signal transducer and activator of transcription, which just means they are the cause to form future genes. Okay. No, now the life-saving interferon can be lethal sometimes. This should be the okay. Again, this slide I'm not going to spend much time on. Just to understand that you have a receptor subunit. In the receptor, the tail of the receptor will be into the cytoplasm, and in the cytoplasm, this receptor tail has some kinases. Just use the term kinases, and somehow they will activate. Okay. So as soon as that the activator comes and binds on the receptor, the downstream kinases gets activated. Once they are activated, they activate another, the STAT. And STAT are the reason that new genes are being formed. So from a modeling perspective, these are the components I need. Okay. So again, we need after this, one thing I should tell you is, there are two types of interferons, as I said, alpha and beta. Surprisingly, alpha can do everything beta does, but beta cannot reverse. Beta cannot perform all the functions of alpha. So those are some of the very strange behavior that kind of guided me to study this particular signaling pathway using some modeling. So as I just mentioned, so this is like your cell, nucleus. So you have your receptor on top. Interferon comes and binds. So you see, it can be any interferon. It doesn't. So this was our question. Either alpha or beta binds. The path is the same. Okay. Everything happens downstream is the same. There is some kinases. They get phosphorylated. After phosphorylation, they kind of phosphorylation means, let us use the term activation. So this tail gets activated. The activated tail will activate this STAT1 and STAT2. They form a complex, and then they start creating the genes. So then we were, talking to some hepatologists, and I will explain the problem next. Okay. Right now, I just set the stage. So at this point, I want to make one comment. That again, body as such, once this gene is formed, more interferons will be formed. Okay, that is like a natural process. So that I had in the previous slide, I didn't read it. Once these genes are formed, more interferons will also be formed. So here comes the question. Then you see, cells will never multiply, right? It will keep repeating. So nature has a natural way of inhibiting the process. Okay. So along with this interferon, some negative feedback factors will also be formed, and they come and bind and stop the process. You see, otherwise you die. So this has to happen. So that natural process is very important, because otherwise cell division will not happen. And that's it. So now the problem. And after this, we will talk a little bit about the hurdles, and I will go back and forth. Okay. So the question, one of the main question was, okay, the subtype can produce, you see, they have a variability. Alpha produces something, beta produces more number of genes. You see, each gene is responsible for a character, which means beta, when it produces a larger number of genes, it's kind of, doing more than alpha. So it's kind of confusing. Then if you look here, you see anti-proliferative actions by alpha. It's different between the two. Alpha is blue, beta is orange. You can see it's different. even though they are sharing the common pathway. So this was, okay, this was one thing we wanted to explain. Can we use some model to explain this? So remember, when it comes from a biologist perspective, you know, some things you hear from childhood, then you know that's right, right? So for example, I drop something and I say it's gravity, you say it's gravity. Do we ever stop to think what it is? No, right? I mean, it takes time. You advance, then you realize, it's not as clear to me anymore. Sometimes we just accept something. So just like that, sometimes biologists may not be even surprised by this, because you know they have been used to, they know that body can do many things and they take it there, and that's how their study goes. So this is where our training, when we link it with a biologist or a chemist, this is where that interdisciplinary study comes into importance. Okay, so never keep a blind eye on, okay, I don't want to learn biology, I don't want to learn. Keep an open eye, because here I have noticed, like I go to physics department when I was in Toronto again. In the physics department, there will be at least 3 or 4 people you can talk to, and they will give you like very fundamental biology. And I was very surprised most of the time. So always keep your mind open and read everything that comes in your way. Okay. So you might find some very interesting problem. So now I will talk about some computational techniques and what are the hurdles when you come to studying systems like this. Okay. So remember, from a physicist point of view or from a mathematical point of view, I can write all of this as differential equations. These are rate equations in chemistry, right? I have one kind of molecules, IFNAR1, it binds with something, I can write a rate equation. Same. So you can see that I can actually simulate every single step here using rate equation. This is where our computational parts come into play. And at the same time, what we call as hurdles, or some kind of trouble come into play. You see how many steps are there. I mean, I have just stopped here, right? There are multiple sequences happening, which means the number of differential equations will just increase, explode. So because of that, usually when you kind of try to simulate this, you will have to choose a small part and you simulate only that. Usually the differential equation is used only to simulate a small part of the signaling pathway. Next is stochastic simulation. I am kind of always into stochastic simulation. There are Monte Carlo or Gillespie algorithms, which means you consider that you understand that two molecules does not bind just like, I'm taking a magnet and attaching it. That is not what happens. There is an association and dissociation constant, and that this is a random process. It's a random event. It's not deterministic like ODEs or ordinary differential equations. So we get that randomness also to play in stochastic simulations. I know that we should have some very good computer geniuses in this group. So I have my cousins cousins who are back in India, and I see how well they do in their bachelor's degree itself. They start doing some kind of little bit of simulations. For students like that who are interested in modeling, there is this software called BioNetGen, which allows you to look into it's free and it's not molecular dynamic simulation. So it's not very hard to play around with, and you can really generate some good results using BioNetGen. So remember, this will allow you to, and BioNetGen means you BioNetGen. So it's kind of a network of biological signals, okay? So what you get here is, if you want to look at any kind of 3 or 4 reactions that come one after another, you can use this software and it's free. And actually it's a good tool to have. You will see, if you check, you will see how many publications are coming out of BioNetGen nowadays. And when we are in small colleges and universities, sometimes funding will be an aspect and we may not have enough time to sit and really code something. So in those kind of situations, to get a jump start, these kind of things will help you. Right now, I'm using something called GROMACS, which is I'm just trying to use. So molecular dynamic simulation, this will be the last computational technique or one of the computational techniques to study these kind of situations. So what is the difference between stochastic simulation like Gillespie and molecular dynamic simulation? So for my purpose, so this depends on what we need, okay? So my purpose was just to answer one question, which I will show in the next slide. I didn't care how these molecules bind. All I wanted to know was, okay, I know the association constants. I know what is the relationship. Can I use this to simulate that system? So for me, that this was enough. But now let us say I am working with someone who wants to have a drug delivery for this particular case. I cannot have this simulation anymore because I'm not trying to find a character. Instead, I am trying to see, okay, which part will it bind so that I can stop it, right? So in that situation, we need something better like molecular dynamic simulations, okay? So again, the question I'm trying to address. So remember, okay, in this case, what are the trouble, like what are the situations that we can land in? One, supercomputers. Many of us don't have access to it, right? We don't have access to it. ODE, as I said, it explodes. Then another thing is training, because we are trained as physicists, we tend to forget, which I will show you later. We even make mistakes because you see, if you have more than 3 parameters and you fit it, you can get anything. You can make a monkey out of 5 or 6 differential equations, but pattern I'm saying. So sometimes we lose track. So that is something I will remind you as we go forward, how to make our simulations realistic and not get carried away. That is what something I will talk about as we move on. So any questions still here? Okay. So next is, this was the question I wanted to address. So let us see how we can model to get an answer. Okay, so the question posed, so imagine a situation like this, you can make it into a modeling or physics part. Think about that. So what happens is, when a patient has a hepatitis, they give interferon treatment. Okay, so 30 minutes of interferon. I'm just showing you how to read a graph. So dark means you can, we may not sometimes have to even explain. When you see a chart like this, so this is called a Western blot. What you see here is some typical protein called phospho or activated STAT. Okay, so after 30 minutes of interferon or 30 minutes of giving the activator, you will see that there is less amount of p-stat for alpha and more for beta. And that you will see this is expected because beta has a higher binding, which means, of course, beta is going to bind faster. So beta, alpha. Now you rest. Rest means you are not giving any more interferon. You stop the interferon. So everything is gone. But the problem was when you give a second dose of the same vaccine, if you give alpha, those patients were not responding after 12 hours. You had to wait more than 48 hours before you can give a second vaccine and to get a kind of response from the person. Beta, on the other hand, it didn't matter. So you see, this is where that question become very important. You have the same shared pathway. Why is alpha, when you give alpha or the one type of activator, why is it not responding? Why is beta able to make a response even though they follow the same pathway? Of course, we can say that is because of the binding affinity, but it is not. If it is just the binding affinity, if I increase the alpha amount, both of them should give me some result, but that is not happening. Now, what are the main challenges in a study like this? You see, this is biology, right? I mean, even though I'm considering it as a molecule and everything, in reality, there is something live going on in the background. So those will kind of affect our studies. So when we go to biological or biophysics studies, we have to be very careful. So now I'm just going to talk about a little bit of characterization of how do you characterize, okay? So again, anyone who is interested in, simulations, just get a MATLAB or, Python free, right? Start simulating differential equation. Use Rangakuta method or something and try simulating, draw some plots, right? And you should know that again, this is called a dose response curve. It's always good to know some terms, So this is a dose response curve, which means here I have the dosage of whatever molecule that I am giving to activate, and how does the response happen. And EC50 just means, I need this for the next discussion. EC50 means at what concentration of my the activator I am giving, I will get half the response, right? Half of the receptors will get saturated. So again, this is the modeling part. So this is where this is what we do. And this is where we can be careful, by the way. So this was our publication. So what we did is we kind of modeled it as a chemical reaction, applied. So again, interferon can attach to this or either of the receptor. Then once one of them receives a interferon, they will start to bind each other and form this complex. So this C is our important part, C or complex. So that complex is what creates the downstream signaling. So as you can see, once the complex is formed, suddenly they activate. So we stopped here, okay? So you see how, even though it's a whole signaling process, when you're doing a modeling, you don't try to simulate every single step downstream. This is what we wanted. Why? Because we are trying to relate the complex to the amount of stat, or the stat is the response. We are trying to relate that together. Okay, so we needed only till here. So again, one thing we needed was, again, this comes from literature review, understanding the system, kind of using one of the negative feedback mechanism to talk about, okay, why is that refractoriness happening? So again, if you remember your chemistry, we use something called detail balance to balance equation. So this is where I'm going to tell you where can we physicists, or we with our kind of training, we can go wrong, okay? So at that time, you see, these kind of models are created with what is known from the experiment, right? So from the experiment, sometimes biology is the information from one cell. Now in the past 5 years, it's really rapidly progressing, but single cell informations are still not available. It's just coming up, okay? So when I was actually looking into this model 2016-17, when I was looking into the model, this was the understanding that interferon can bind to either. So this is where I'm going to be more cautious as we go on to tell you, when we start a new research, what are the topics, what are the things that we should pay attention to. Oh, I think I'm running out of time, so let me go fast now. So again, as you can see, there is a model, right? This is called detail balancing, and we realized that from some experimental thing, we saw that actually there was some mistake, one of the detail balance, or what we thought should obey, was not being obeyed. Okay, so we looked at some experimental data available, and something was not matching. So what we thought was, maybe that experiment is wrong, and we kind of use detail balancing to model our system. Anyway, bottom line is, with the 2-receptor model, when you kind of try to do some simulations using stochastic simulation, you will see that it's a natural, if it is a single receptor, okay, this is why I said I don't use the term evolution lightly anymore, because if this model was with a single receptor, this kind of a differentiation that we saw was not possible, which means the cells will have to create different type of receptor and different type of activator to get each of these genes. But somehow that's not how it is. You have 2 or 3 receptor, which will combine together, compete against each other, and form a pattern. Okay. So this differential response is actually a natural consequence of a two receptor subunit. So I had Salino, we of course, we cannot do without some equations. You have to show some equations. So we have that. And again, we can just write some equation, and actually we even matched it with some experimental results. I'm kind of rushing through it, but we could match it with the experimental result. But bottom line is, with a two receptor subunit model, we were able to come up with this fact that the cells can differentiate between the different activators because it is a two receptor subunit. They cannot achieve the saturated maximum. So that was the bottom line of our study. Again, I'm just going to come to a reduced model and tell a little bit more. So here you see what are the mistakes, right? Or what are the difficulties? So that is what after conclusion I'm going to say. So this is what we concluded. We thought, okay, we found out a very well-reduced, well-calibrated model. We have some, very small number of measurable parameters, which means we didn't use too many parameters to model the equation. You see, everything came from experiment except one. And we also said that, interferons can be distinguished based on their, so we came up with some things and it was good. We could match it up, everything is good. And luckily for us, whatever mistake we made didn't affect our publication, or because of that, we didn't publish some of the results. So this was my team. Now, revisit. So recently, I started revisiting my models. And then I realized, so now, things are improving. 2 years, things have improved drastically. There are some pathway models which will actually look at all the publications in the world. Okay, so it's actually from Science Direct. It will look at all publications and give you a network analysis connecting, so that means modeling becomes very easy. So here you will see that actually, though there was one of the reactions that I modeled was actually not possible, and that was found only in 2020 or something. So that was new. So that the reason why the detailed balance was not working had a reason that was not biologically happening. Okay, so that was why detailed balance, but at that time that was the understanding. So you see, these are the kind of problems that comes when you go into biological or something like complex systems research. You will see that there is the interacting environments and there is a lot of unknowns. So from a modeling perspective, we might start putting together many numbers and equations and we will get the results. So this is where you have to be careful. So when I was in my bachelor's degree, I don't remember the person, but I still remember the cow. I still remember it. I attended a talk, and I don't know if any of you have seen this cow before. When a physicist, a theoretist start a research, you want to draw a cow, you will draw a circle, right? So yes, I had to start from a circle, but the idea is, you can look for problems around you like this, start drawing your circle slowly, and from there you start working on it, right? And try to improve it to make it a real cow. Now, my real goal, of course, is, so as a physicist, again, my thing is to apply network analysis and some of the, thermodynamics models to figure out how these signaling pathways are interconnected. Now, since we are discussing hot topics and, when COVID came, so I just want to, my last slide, I just want to tell you, like, there is a very emerging hot field, even for us physicists coming up, health disparity research. During COVID, they started saying, like, of course, I'm in United States now, so I'm looking at the data here. This is definitely true for any place, right? We always think that when there is health disparity, it is because of, economic situations or social status. It may not be just that. It can be gene expression levels, which means, places where there is a prevalent malaria, for example, some of our genes will be switched off. So that can cause some other problems later on. So actually, again, we can use physics, modeling, network theory to actually study these things. So that is my current focus. So I just, again, what will be the hurdles? Again, 1, data. 2, we really don't know how to model it, right? So you have to come up with something. These are all very, very new emerging fields. So I always, now I have realized that with time, and so I was reading my Bible, and this verse I was reading recently, and I thought I should close with this, and I will take any questions if you have. I think I ran over time. Sorry about that. Could you all hear me? So any questions? If you say yes, you have to ask a question. That is my rule in my class. Any questions for me, even general, or I think anyway, we are run out of time. So
Thank you, ma'am. It was a great pleasure listening to your valuable insights on the topic. I strongly believe it was informational to our audience present here. Now, the session is open to questionnaire. You can either raise the hand, unmute yourselves and ask your questions, or you can out your questions in the chat box and we'll read it out to Dr. Chitra Naik.
I see there is one question. Can the statement everything? Yeah. So again, everything came from a single cell be validated. This is why I said I made my statement clear multiple times, because that the fact that can I really talk about it all came from a single cell? I really don't think so. I have been looking for this. I have been talking to evolutionary biologists for a while about this. At least from my part, I never understood it. That is what I should say, and I'm glad I did not. So I don't think anyone can show it, but let's see. No, my answer will be no. If anyone is telling you that there are some new technology which can, I would say no, still, because I don't think right now, because I have been following this quite from 2 perspectives. I've been following this for a while now. Okay, why did I get it? Actually, I didn't. I never thought I will like biology. I actually went to do my first postdoc because that person was doing something in bacterial cell, and as I said, he was looking into some wave oscillations. So I read his paper. His name was Andrew Rutenberg. He's my first postdoc boss in Dalhousie University. Amazing person. He was from Princeton, and then he did his PhD from Princeton and Bell Labs. He's a statistical mechanics person. Every 3 years, he will change subject, which was something new to me. So now I also change topics. I try to, when you respect someone, you try to do that. So right now he's working in something of old age. So when I went there, his interest on that topic was gone. So I read some of his papers. I wanted to work on it. I wrote to him, I want to work on it. But when I went there, he was bored. So he wanted to move on to something called how do cells transport across bacteria. And once I started reading, so I started okay, this will be one project of mine. But then you understand that, okay, you look at the sky, but there is more when you look into a bacteria. Was kind of eye-opening for me. So then these small organisms are so difficult, you don't even know about them. Of course, you know a lot about them, but you don't know anything about them. So, okay, I see biophysics opportunities. I think it's a very hot topic, as you can see. Earlier again, bringing back to Dr. George Verghese, he said you don't get funds, right? So the one way to get fund is to do research that really matters, right? I'm not saying fundamentals. I am a fundamental science person. But when money becomes a constraint, where do you invest in, right? You invest in things that brings out some results for the general community. So here they tell us, like when you are writing for a grant, make sure that there is an outreach aspect. You are using taxpayers' money. So when you are using taxpayers' money, there should be some way they should be also getting some benefit, right? So yes, for that reason, I think biophysics has a lot of option, everywhere it is coming up. And I think we should not separate science. We should keep it as science, right? Science and math is the language to talk about science. I would rather say something like that. Yeah, we have a lot to do actually. Like, sometimes you have to, oh, where is best? I don't know. Like, that is a hard question, Rohit. I kind of like, again, we have to incorporate research in our bachelor's degree onwards, maybe even before, because I work with school students here and, for science fair, science Olympiad and all. So you kind of start making them do things before. And as I said before, even during my postdoc, I did not have the courage to ask a question which just means that it brought me down so much, right? Because you didn't ask the question in the right time. So for that reason, I like it here. But maybe 10, like what, 12, 13 years I'm here, it might have changed in India a lot. Now you guys may not be as scared as I was. So, but I know many from my time, my generation had that problem. We won't ask a question. Oh, do some internship, Chaitanya, like, do some internship, get like, right after masters, I went for a small short spring in IASC. I was in IAA for a while. Whatever little options you get now, and talk to people, anyone who comes, maintain the contact, send them a note. Sometimes, in real busy times, we might, I have done it. I'm still trying to find one student who emailed me 1 year back from Kerala. I somehow missed that email. I couldn't remember the name. I went back, I couldn't find her. I still regret. So if you don't get a response from someone, just send another email. Yeah. So, that's the way to keep in touch, right? And try to get some internships, always. And there are a lot of opportunities in India, like IASC, IITs have some internship opportunities. So apply for that. Go somewhere in summer or work with your faculty. You start one, I said BioNetGen, download it, put some equations. There is something like, COVID. How does COVID bind to your receptor? There is a receptor called ACE2, and COVID binds. Take that as your problem, nothing else. You are learning a new thing, right? You are learning something called BioNetGen. You are understanding how coding works. So that's it. And yeah. Okay, I think you may not even have your tea time anymore.
Ma'am, can I ask you a personal question?
Oh yeah, definitely.
Ma'am, how did you get into that university? Because I would like to study from abroad after my graduation. I would like to do some internships there. And
I did my all my education was in India, by the way. So my postdoctoral, no one considers. I mean, it's kind of continuing research, right? So till my PhD, I didn't even have in my mind. I didn't even have a passport. So my everything was in Cochin University. But at the same time, if you want, I know students from, like, I was in Madras for a while, so I know students from Madras University. They used to apply, like, GRE and come on, like, once you go for, but mostly it will be after masters. Our bachelor's program is not recognized here because my students who do bachelor's is 4 years, whereas in India we are 3 years, right? So you have to complete a bachelor's and masters, then do GRE. You can come, you can apply, and then you might, depending on your strength, you might have to do one course, 1 year course, and then go for your PhD program. That's the usual scheme. So GRE, mostly that's the one way of coming here, especially with COVID. I know it's pretty tight, but that's the real way of coming. But again, I did all, and I know another faculty here, he also did everything in India. We have good system in India for that matter. So you do your PhD from India, you have a lot of possibilities. And I think like most of us, tomorrow your speaker Hari Krishnan, he did PhD in India. Most of us did our PhD in India and then moved out. That's nice questions. Thank you.
The time for questionnaire has come to an end. Thank you, ma'am, again for your time and words. Next, we have the paper presentation session chaired by Dr. Annamma John, Professor and Head, Department of Physics, Marine Vanish College, Trivandrum. Dr. Annamma John completed her MSc, MPhil and PhD from the University of Kerala and has a teaching experience of about 23 years in total in both St. John's College, Anchal and Marine Vanish College, Trivandrum. She has done her specialization in vibrational spectroscopy. She did a major project that has been funded by KSCSTE, Government of Kerala, during the years 2013 to 2016, titled Synthesis and Characterizations of Material for Dielectric Resonators and Solid Oxide Fuel Cells. She has done about 34 international and 1 national research paper published in journal and has to her credit 4 international publications and 1 national publication in conference proceedings. She has around 39 publications and 343 citations. I'm inviting Dr. Annamma John to carry on with this session. Over to you, ma'am.
Good morning. Am I audible? Can you hear me? Can you hear me? Am I audible?
Yes, ma'am.
Okay. So good morning. First of all, let me congratulate the Department of Physics, Christ Nagar College, Maranallur, Thiruvananthapuram for organizing an international conference on recent trends and hurdles in physical sciences research. Actually, these two words, the phrase recent trends and hurdles is a relevant combination of words. The study of recent trends and hurdles in a particular field is actually a positive approach to excellence. And I'm happy that the college department and the college has taken such an initiative. And it's my pleasure to chair the first paper presentation session of this event. I express my heartfelt gratitude to the management and the organizers for giving me this opportunity. And in this session, we have 4 presentations, and each presentation is scheduled for 10 minutes with 2 to 3 minutes for discussion. So participants listening to the session, please be free enough to raise your queries after each presentation. Okay, let us go to the, move on to the matter. The first presentation is by, I call upon Ms. Akhila Murali J to present her paper titled Heteroatom Doping in Folic Acid Derived CDOTs for Enriching Photoluminescence Properties. Akhila Murali?
Yes, ma'am. Can you hear, ma'am?
Yes. You can start.
Ma'am, is this slide is visible?
Yes.
Okay, ma'am. Good morning, all. Myself, Akhila Murali, research scholar from Department of Physics, University of Kerala. Today, I am going to present a part of my research work entitled Heteroatom Doping in Folic Acid Derived CDOTs for Enriching Photoluminescence Properties. My presentation includes Excuse me, ma'am. I can't go for the next slide. Sorry for the delay. My presentation includes a brief introduction on carbon dots, objectives and motivations behind this work, synthesis procedure and characterization techniques, and the results obtained with their corresponding discussions. Fluorescent nanoparticles light up the otherwise invisible process that make our biology tick, or in the case of diseases like cancer, that make it go away. Some of the most powerful particles of this type are so-called carbon dots. They tend to be easier to fabricate and more compatible with our biological makeup than other similar nanoparticles. Carbon dot, or abbreviated as C dot, is a comprehensive term for various nano-sized carbon materials. We can describe these C dots as a polymer-like structure with sp3 hybridized amorphous carbon cores that contain small domains of partially sp2 hybridized carbon atoms. These carbon dots mainly include graphene quantum dots, carbon nanodots, and polymer dots. The GQDs possess one or a few layers of graphene and connected chemical groups on the edges. At the same time, these carbon nanodots, that is the CNDs, are always spherical and they are divided into carbon nanoparticles. The polymer dots, or the PDs, are aggregated or cross-linked polymers prepared from linear polymers or monomers. Nowadays, these carbon dots have achieved rapid development because of their excellent properties, including low toxicity, easy to synthesize, superior stability, adjustable excitation and emission wavelength, and rich surface functional groups. Unfortunately, they also tend to be fairly dim, emitting relatively few photons versus the amount they absorb. Recently, researchers report a new method that vastly improves the quantum yield of carbon dots, reaching a high efficiency of 94.5%. What's more, the bright dots intrinsically target certain cancer cells, paving the way toward the better cancer diagnostics. The team Loyo et al. Formed the particles through a simple one-step method involving single ingredient called the folic acid. We know that folic acid is one of the B vitamins which consists of rich oxygen and nitrogen functional groups. Since it helps in regenerating healthy cells, folic acid is one way to track some cancer cells in the body. Over the past few decades, most of the research target the photoluminescence properties of C dots. But the universal origin of the fluorescence in the carbon dots still remains as a mystery and is a subject of debate. The major investigations include optical selection of differently sized nanoparticles, that's the quantum effect, and the surface states, and the third one is the molecular fluorescence. It is relatively easier to modify the surface of C dots, thereby tune the color emitted, because it is a general belief that the color originated from the surface state or the surface defects. Heteroatom doping is a powerful approach for adjusting the intrinsic structure of C dots. The introduction of heteroatoms can change the surface structure and electron distribution in these carbon dots, which is related to the energy gap between the HOMO and LUMO, thus affecting their fluorescent properties. The commonly used dopants are nitrogen, sulfur, phosphor, boron, silicon, etc. In the present work, we used the rarely reported fluorine atom and the commonly reported nitrogen atom as the heteroatom dopants because they are the immediate neighbors of the core carbon. Here, the folic acid derived carbon dots, which is abbreviated as FCNDs, were synthesized via hydrothermal treatment at 180 degrees Celsius. Ammonium fluoride and ammonium hydroxide are added in the synthesis procedure for the synthesis of fluorine doped carbon dots and the nitrogen doped, which is termed as FFCND and NFCND samples respectively. The synthesized C dot system was studied using X-ray powder diffractometry, Fourier transform infrared spectrometry, Raman spectroscopy, X-ray photoelectron spectroscopy, UV visible absorption spectroscopy, photoluminescence spectroscopy, lifetime measurement, and quantum yield measurements. To preserve or to tune the optical properties of C dots, it is necessary to understand the structure of C dots and their fluorescence origin. Here, the broad reflection in the XRD pattern corresponds to amorphous carbonaceous materials, and the peak at about 28 degrees is associated with the 200 crystallographic plane of the graphite. Compared to the graphitic structure, The shift of 2 theta from the reported 25 degree may be resulted from the incorporation of more oxygen containing groups and decrease in the sp2 layer spacing during the carbonization process. Also, the Raman spectrum, the deconvoluted Raman spectrum here shows two peaks corresponding to the D band and G band. In general, the intensity ratio ID by Ig is used to analyze the structural order. Here, the relatively high intensity of D band indicates the presence of diamond structure, that is the presence of sp3 defects within sp2 clusters. Here, the third figure, the SEM image of FCND shows that the asynthesized FCND, that is the folic acid derived carbon dots, have a size range within 10 nanometer. This absorption spectrum, their absorption spectrum of C dots showed two distinct peaks at 235 nanometer and 278 nanometer respectively. The absorption band at 235 nanometer corresponds to the pi to pi star transition of sp2 carbon, and that of the 278 nanometer arises from the absorption of aromatic pi system of C double bond O. The photoluminescence spectrum of the carbon dots was located about 460 nanometer, or we can say that it is concentrated on the blue region of the EM spectrum with an absolute PL quantum yield of 10.60% obtained from the integrating sphere method. With this understanding of the structure and fluorescence of the bare C dots, the effect of heteroatom doping on the structural and optical properties of folic acid derived C dots was investigated. The FTIR spectroscopy was used to unveil the heteroatom doping and functional groups present in the carbon dots. In the FTIR spectrum of C dots, that is the FCND, that is the bare carbon dot, FFCND, that means ammonium fluoride doped folic acid derived carbon dot, and ammonium hydroxide doped, that is the NFCND. Many characteristic peaks are observed, which corresponds to mainly OH stretching, NH2 stretching in the range 3,100 to 3,500 centimeter inverse, and also there is a C double bond stretching around 1,700 centimeter inverse, etc. And also the semi-ionic CF and covalent CF bond in the range 1,000 to 1,300 centimeter inverse indicates that the fluorine get bonded with the carbon. Next figures the Raman spectrum. The Raman spectrum of the samples excited at 735 nanometer laser shows two prominent peaks into range 1,000 to 2,200 centimeter inverse. The deconvolution of the main peak using Lorentzian function gives two peaks corresponds to D band and G band, which we have seen earlier in the slide. The next peak, that is the 1897 centimeter inverse, it is associated with the N heterocycles and phenyl groups associated with the folic acid. That is, the structural memory of the carbonaceous precursor is retained, which is the unique property of the carbon dots. To further elucidate the structure, X-ray photoelectron spectroscopy was also performed. Here, the high resolution scans shows that this material, we select only one representative material, that is FFCND, and it is composed of mainly 4 elements: carbon, nitrogen, oxygen, and fluorine. And the high contents of carbon atom indicates the carbonization occurred during the synthesis. And the table shows the binding energy peaks and the corresponding atomic percentage of all these elements. Now, how the heteroatom doping affects the luminescence properties of the folic acid derived carbon dots. We know that when we discussed the earlier part, the folic acid derived carbon dots, that is the bare carbon dot, emit PL emission, or they show strong luminescence in only blue region. Here, we doped it with the fluorine and nitrogen. The ammonium fluoride doped sample, that is the FFCND, shows an absorption spectrum identical to that of the bare carbon dot, but there is some shift in the absorption bands, while the pi to pi star transition is absent in the NFCND, that is the ammonium hydroxide doped sample. That is, there is change in the electronic levels or the electronic transition levels. This variation in electronic levels is supported by the photoluminescence spectrum. Here we can see that the heteroatom doped C dots exhibit strong green emission at a higher wavelength excitation, which is absent for the earlier bare C dot. It is generally believed that The green luminescence from C dots originate from the defect states with the oxygenous functional groups. And both these samples shows a quantum yield of 5% and 5.43%, which is calculated using the integrating sphere method. And the last figure shows the photographs showing the color of the carbon dot solution we have prepared in the daylight under UV light excitation and at a 460 nanometer excitation. Here we can see that the blue color in the UV light and there is a green emission in the higher wavelength excitation. And this PL spectrum clearly shows the redshift of the emission wavelength of the carbon dots by the heteroatom doping. There we can clearly show that this straight line is deviated from the peak of the three samples. That is, as the heteroatom doping is doping is produced in this material, there is a shift of the emission wavelength. That is why it has happened, because when we look for the nitrogen material, nitrogen is an n-type impurity that provides excess electrons to the system, which rises the Fermi level and changes the optical properties. Similarly, the fluorine being the maximum electronegative atom, the fluorine moiety strongly absorb the adjacent electrons and increase the separation between charges. This increase in the extent of pi electron system reduces the band gap, that is the narrowing the band gap between the levels, which causes the emission in the longer wavelength region. Here we plot the PL emission spectra for two excitation wavelengths, that is the 360 nanometer and the 400 nanometer only. Now, to acquire a deep understanding about the luminescence mechanism, the PL dynamics of the carbon dots were determined. The decay curves were fitted with the bi and tri-exponential decay functions. That means the luminescence of these carbon dots, that's the bare carbon dot, and the representative sample, that is the fluorine doped FFC and D, were consist of several emission bands from specific emission states. That is, we can evident from the tri and bi exponential function. And also the decay time is calculated to be 7.72 nanosecond and 6.63 nanosecond respectively. In short, we have successfully synthesized folic acid derived C dots using simple hydrothermal method. They show strong fluorescence in the different excitation wavelengths. While we introduce a heteroatom, the heteroatom doping causes green luminescence from carbon dots at a higher excitation, which may arise from the oxygenous functional groups and the electronic state variation. The presence of several emission states within a single emission center is confirmed by the TCSPC result, that is, by the multi-exponential decay fit. Thus, we can say that the heteroatom doping changes the surface structure and electron distribution in C dots, and which in turn results in a redshift in the PL emission wavelength. Thank you.
Any questions from the participants? Do anyone have any questions? Akila?
Yes, ma'am.
Since no one hearing the presentation is asking you a question, so I thought I'll ask you one question.
Okay, ma'am.
About the Raman spectrum, no, the IR spectrum you have mentioned, which is the nitrogen-hydrogen band, the CO band, everything is mentioned. And in the Raman spectrum, you have named it as D and G band. What is this D and G band?
Ma'am, D band corresponds to the diamond nature of the diamond band, that is the sp3 nature, and the G is the graphitic band.
Okay, okay. Oh, oh, okay, correct. Now only I understood. I was thinking of what is this D and G band. Okay.
No, ma'am. Because the basic part is the graphitic structure, so it is we are named it as D and G band from the graphitic structure.
Okay. Okay, it's a good work. Anyone asking her question? Anyone has any doubt about this work? It's an excellent work. It's a photoluminescence property of folic acid. It's really a good work. Anyone has a question? Okay, thank you, Agila.
Okay, thank you, ma'am. Thank you so much.
Okay, we'll move on to the next presentation. It's by Miss Indulakshmi J. And she is presenting the paper on monomer or dimer degradation in methylene blue using iron oxychloride as photocatalyst. Miss Indulakshmi, are you there?
Yes, ma'am. Am I audible?
Okay. Yes, you are audible. Okay, you can start your presentation.
Okay, is the slides visible?
Yes.
Okay. So good morning, everyone. Our work is entitled Monomer Dimer Degradation in Methylene Blue using iron oxychloride as photocatalyst. So here we have an outline of this presentation. So the background of the study involves one of the major issues that we are facing today is the shortage of potable water. We have water availability, but that water is getting polluted day by day. And when we consider the statistics of water pollution, we can see that the textile industry and the organic dyes that are released from the textile industry is contributing to about 1/5 of the total water pollution. So we need a solution for this. And we can see that these organic dyes, they have both environmental and health hazards. Environmental hazard means these dyes are covering the water bodies and have direct effect on the aquatic water life. Also, they are entering the food chain, and also these dyes can affect the human bodies, that the central nervous system or other organ systems of human bodies. So it is a need of the hour to remove these organic dyes from the water. So how can we remove these dyes? There are various processes available for this, but these processes like adsorption, reverse osmosis, all of these processes have one or another pitfalls or shortcoming, or they are specific to certain applications. So here we are choosing another method, that is photocatalysis, to remove these dyes from water. So what is photocatalysis? Photocatalysis, we know that it's the employment of a light activated material to alter the kinetics of a reaction. So here in employing photocatalysis to remove the organic dyes, we have a material which may be a semiconductor, and this is photoactive. So when light of suitable energy, that is the energy, light having energy higher than the band gap of the material, when that kind of energy is incident on this material, what happens is that electron hole pairs are getting liberated. We can see in the diagram, there are electrons in the conduction band and holes in the valence band. And we can see the holes will oxidize the reactants to form products, whereas the electrons will reduce and form products. So this can degrade the organic dyes. So choosing the material as the photocatalyst. So we are focusing on two-dimensional materials because they have maybe layer structure and higher surface area. So this would be beneficial to improving the photocatalytic activity. Further, we are choosing an iron-based material because iron is available in abundance. So we can have a cost-effective method of photocatalysis. So here, our material is iron oxychloride, which is a layered material. And we can see the crystalline structure in the slide. And also, the model organic pollutant that we have chosen is methylene blue, which is commonly found in textile industries. So our objectives will be to synthesis and characterize FeOCl and to study the photocatalytic performance, including the monomer dimer kinetics. So here we have the synthesis technique, and we can see that we have synthesized the material using the solid phase pyrolysis of ferric chloride hexahydrate. And following the steps are the ultrasonication and vacuum drying, and we will obtain nanostructured FeOCl, which was characterized by various characterization techniques. So here we can see the X-ray diffraction pattern of the material. So here we can confirm that our material is formed in pure phase. No other impurities were formed. And from in the XRD pattern, we can see the first peak is the major peak, and here it is a 010 peak. So the 010 peak means that if we calculate the d-spacing corresponding to this peak, that d-spacing we have obtained it as 7.91 angstrom, and that d-spacing is actually the distance between the FeOCl layers. I have already told that FeOCl is a layered material. So this d-spacing, this is actually the distance between two layers, and we can see that the green atoms, those are chlorine atoms, and the van der Waals radius of chlorine atom is less than 2 angstrom. So the d-spacing is almost 4 times than the van der Waals radius of chlorine atom, and we can confirm that this is indeed a layer structured material. And we have the scanning electron images, and this is also a layered structure with an enhanced surface area. This is a peculiar structure. It's a fibrous structure which are agglomerated to form like a grass-like substrate. So this will also enhance the surface area, which can have positive contribution towards the photocatalytic activity. So to study whether this material can be used as a photocatalyst, we need to know the band gap. So we have the UV visible spectra here. We can see the material is absorbing the absorbance spectra. The apparent absorbance is shown in figure 4b. We can see the material is absorbing in a broad range, and we have calculated the band gap of material to be 1.70 eV. That is given in the figures 4c. We also study the thermal stability, and we can see that material is stable up to a temperature of 445 degrees Celsius. Also, we have the Raman spectra here. From the Raman spectra, we can see that there are vibrations from the FeCl bonds and FeO bonds. The FeCl bonds, actually, there are 2 peaks. This may be because in our material, there are 2 different iron chlorine coordinations. So these 2 different coordinations are giving rise to 2 different peaks. And also we have FeO bonds, and those also due to the FeO stretching vibration. There is an additional FeOH bond is occurring. This is actually occurring because in layer structured materials, water intercalation may be possible. So these water, here we have Fe3 plus and Fe2 plus states in the material. So this interacts with this iron atoms, and they are forming FeOH bonds. So we are moving on to photocatalytic studies. Here we have used methylene blue as a pollutant, and we are adding the iron oxychloride to this methylene blue solution. Then first we will place this system in darkness for 30 minutes. This is to make an absorption-desorption equilibrium to occur. And after the 30 minutes, we are exposing this to direct solar radiation. So then what will occur is the photocatalytic degradation of the material is occurring, and decomposition products will be formed. So to analyze the decomposition, we will consider the time-dependent UV visible spectra. So here we have the figure 7. This is the time-dependent UV visible absorption spectra of methylene blue. We are analyzing the concentration of the solution by taking samples with time. So from the figure, we can see that initially the methylene blue concentration is very high, but it is decreasing with time. That means that the concentration of methylene blue is decreasing. So we can calculate the variation in concentration of methylene blue due to photodegradation, which is given in the second figure, that is figure 8. We can see that there is a subsequent fall in the first 10 minutes itself, and the concentration is decreasing, and we can see the last dot is corresponding to a value lesser than 0.2, which means that 87% degradation is occurring in the first 30 minutes of activity, which is a very good value for a novel photocatalyst. And we need to study the kinetics of this reaction. For that, we have employed a pseudo second order model. And the pseudo second order fit is given in the figure 9, and we can see we are getting a good fit. And the apparent rate constant, we have 0.10 plus or minus 0.01 per minute. So usually in photocatalytic reactions, we have pseudo first order model, but here we are getting a pseudo second order model. So why is this happening? Also, something to substantiate the use of pseudo second order model can be obtained from the concentration variation. That is the second graph. We can see in the first 10 minutes, there's a large scale decrease in the concentration. But with further time, that decrease is not so large when compared to the first 10 minutes. So this means that there is a dependence on the concentration of methylene blue, and that's why we are employing second order kinetics. Another reason for second order kinetics to happen is the presence of dimers. So we know methylene blue is an organic molecule, and these organic molecules, they can form dimers or tetramers. Further polymers are formed. In the case of methylene blue, there are both monomers and dimers. So in the absorption spectra of methylene blue, we have seen the first figure, figure 7, we can see there is a major peak around 654 nanometer. This is due to the monomers. And the shoulder peak, this is occurring because dimers are contributing to this peak. So we can study the monomer and dimer concentration in the methylene blue by analyzing these 2 peaks. So here we have used, we have the table here. In this table, the first we have the peak position, peak area, and the concentration. And inviting your attention to the last column, which shows the monomer dimer ratio. So the first value is 0.46. This is the initial value in the pure beryllium blue solution. And after 30 minutes of dark degradation, the value is not much changing. This because this suggests that both monomers and dimers are getting absorbed in equivalent amounts. But after 10 minutes of degradation, we can see that this value decreased to 0.40. Why is that happening? This means that the concentration of monomers are decreasing. That means there is a preferential degradation of monomers in the first 10 minutes. But after, but with subsequent time interval, we can see in 20 minutes, the value is increasing up to 0.61 and is remaining nearly constant with further light exposure. So this process can be explained like this. So first, in the first light exposure, monomers are getting degraded because they are not, they're simple structures, so they are directly getting degraded to form decomposed products. But the dimers, they are complex structures and are not getting easily degraded. But when the monomers are removed, the concentration of methylene blue is decreasing. So the tendency to form dimers is also decreasing. That is why the dimer concentration is decreasing in the further exposure. And beyond that, we can see both are almost removed in equivalent amounts, which suggests that in the presence of iron oxychloride, there is not much preferential removal of monomer and dimer. So we can conclude that in this work, we have synthesized agglomerated fibrous FeOCl nanostructure having a band gap of 1.70 eV and having a very good photocatalytic activity of 87% degradation occurring in 30 minutes, which is a very good photocatalytic activity. And we have used pseudo second order kinetics, which is attributed to the formation of dimers. And both dimers are monomers are degrading in almost equivalent amounts. And we can say that the layer structure and narrow band gap favors the photocatalytic activity. So thank you.
Thank you, Induja Lakshmi.
Yes, ma'am.
I think I have called you for the presentation as Indu Lakshmi, no?
Yes, ma'am. Yes.
Yeah, sorry. Now only I recognized my fault. Anyway, the session is open for questions. Does anyone have a question? It's really a very interesting topic, this photocatalysis, where we have many applications. Anyone? Anyone have a question? Okay, then I'll ask a question just to have a discussion session. I'll ask you a question. You have said the band gap is 1.7 electron volt, and how do you connect to this band gap with the photocatalysis activity?
Actually, ma'am, to choose the photocatalyst, we need to have a material having a lower band gap, because if the band gap is less than 2 and all, the energy is lesser. So a light of energy higher than that can excite the material. So if we are having a material having a band gap greater than 2 and all, we can excite that material only by using light having energy higher than that value. So that will limit the application. So here in this material, we have 1.70 electron volt that corresponds to an energy of red light. So that means that even the violet light can excite this material. So if it is a higher band gap, we can't do this. That is why we can employ the full solar radiation, because most of the photocatalysts, we are having materials that are excited by UV light. So that will limit its application. That is the advantage of this material. This is a comparatively lower band gap and can be used for wider applications in terms of photocatalysis.
Okay. You have presented photocatalysis property, and actually I have read somewhere that the materials which are suitable for photocatalysis have a low photoluminescence property. Emissions are should be very low. I think I have read it somewhere. Am I right?
Ma'am. Yes, ma'am.
Photoluminescence.
I'm not quite sure about that, but our material is not showing photoluminescence activities. But I think there have been materials which show PL emission as well. Actually, I am not aware about such a correlation. I shall read into that. I can't.
Okay. I'm also not that sure, but I have heard it and I have read somewhere that It's you don't study the photoluminescence property, no?
Yes, ma'am. I have actually studied this material is not having any photoluminescence.
Yeah,
It's
yeah.
actually an indirect band gap material, so it's not showing any.
Okay, okay. I think that's why it's giving us such a good photocatalytic activity.
Yeah, maybe.
Okay. Anyone listening to the presentation have
Yes.
a question? Okay, okay, ask.
Can you hear me, ma'am?
Yes, yes.
Okay, dear Induja Lakshmi, the presentation was very nice. And the compound is metallic compound, no?
Ma'am, it's
Right?
actually it's not metallic, it's actually a semiconductor, which can be class. It's almost a semiconductor.
Yeah, semiconductor. That was my question. The band gap was coming in the range of semiconductor. It comes to the nano size, the compound has become showing some semiconductor nature. No.
Yeah, it's actually, this material is actually a Mott insulator. This is, by this band gap, this falls under the category of a semiconductor, but other properties just slightly different from a semiconductor. So it's actually a special class of materials called Mott insulators. Actually, the conductivity values will be lesser.
Okay, okay. Did you take the sample at a different time and test it for the concentration?
No, ma'am, actually, we have only one sample used. Is that what you meant? Sample at different times means?
Yeah, as the time goes on, the degradation happens in different percentage, no?
Yes, ma'am. Ma'am, actually, are you talking about the degradation means?
Yes, yes.
This figure?
Yeah.
Actually, ma'am, we are exposing this to sunlight and then studying the UV spectra at constant time intervals. So we have actually studied this up to 1 hour. Even up to 3 hours, we have studied. But the best activity is occurring in the first 30 minutes itself. This is actually giving 87% degradation in 30 minutes. That's why we have shown the first 30 minutes here. Ma'am, actually, could you clarify? Is there any? I am actually not clarified with the question.
I'm asking about if you are taking different sample of this constant time interval and take the UV.
No, ma'am, actually, we have done with you only one sample. We have repeated the same experiment.
Okay,
That's all.
That was my question. Thank you.
The errors were calculated with the same material and the same experiment with a different set of experiments. That's
okay, that was my question.
Thank you, ma'am.
And you stopped at 30 minutes because the curve has flattened, no?
No, ma'am. Yes, ma'am. Actually, we have studied it up to 3 hours, but the major degradation is occurring in the first 30 minutes itself.
Okay, Induja, a nice presentation.
Thank you,
and wish you all the best.
Thank you, ma'am.
Okay. Now we'll go on to the third presentation of the session. May I call upon Miss Geeta R. She'll present her work. Ma'am? Ma'am, whether I am audible? Yes, you are audible.
Good morning to all. Myself, Geeta. I am going to present a part of my research work on DFT vibrational spectroscopic and molecular docking studies on 6-methoxy-8 nitroquinonium picrate, a novel cervical cancer drug.
Geeta,
yes, ma'am.
You go to slide presentation.
Yes, ma'am. Yes, ma'am.
Slideshow you go.
Whether it is visible, ma'am?
Visible, but you are still with the initial page. Can't you go for slideshow?
Yes, ma'am. Yes, ma'am. Introduction. The quinoline compounds have a benzene ring fused to a pyridine ring at two adjacent carbon atoms. The quinoline compounds are widely used as the parental compound to synthesize the molecule with medicinal benefits. Compounds with quinoline motifs have a variety of chemotherapeutic activities, especially antibacterial, anti-malarial, antifungal, anti-cancerous, anti-tumor, anti-diabetes, etc. This quinoline found their application as solvents for terpenes and resins, polymers, catalysts, for preservatives, and corrosion inhibitors. Quinoline cores are found in numerous organic compounds such as chemotherapeutic agents, analytical reagents, and alkaloids. So among all these quinoline compounds, I have selected 6-methoxy-8-nitroquinoline picrate as it has more oxidizing properties as well as potential anti-cancerous application. Literature survey reveals that the structural characteristics and vibrational spectroscopic of the pharmaceutical compound has not been studied to the best of our knowledge. In the present study, an extensive DFT calculation, vibrational spectroscopic and molecular docking studies on 6M8-NQP was carried out. The FMO analysis, UV visible spectral analysis, NBO analysis, and Millikan atomic charge distribution analysis of the compound were also studied. Computational details, the global minimum energy molecular structure was optimized by DFT B3LYP method with CCPV-TZ basis set using Gaussian 09 program. The molecular structure and FMOs were visualized by GaussView 0 by visualizing program. The FMOs first order hyperpolarizability and NBO calculations were carried out by DFT B3LYP method with CCPV-TZ basis set. The UV visible spectrum was studied by time-dependent DFT B3LYP method using CCPV-TZ basis set using ethanol as a solvent. The NBO calculations were carried out using NBO 3.1 program as implemented in the Gaussian 09 program. Millikan atomic charge and local reactivity values were calculated using Millikan population analysis with 631G DP basis set. Material synthesis and optimized structure. The analytical grades of 6-methoxy-8-nitroquinoline and picric acid were taken in equimolar ratio. The reactants were dissolved in chloroform and acetone solvents. Brown color crystals were harvested using slow evaporation technique. The optimized structure is shown in the figure. The molecular structure of 6M8-NQP molecule has 39 atoms and 110 normal modes of vibration, which belongs to the same symmetry species. The important vibrational frequencies, IR intensity, Raman scattering activity, reduced mass, and force constant values were calculated for the more stable optimized structure of 6M8-NQP molecule. Results and discussion, FTIR and FT Raman spectral analysis. The CH stretching vibrational modes is normally observed in the region 3-100 to 2-800 cm raised to -1. Here CH stretching vibration were calculated as 3288, 3278, 3245, 3237, 3222, 3225, 3201, 3175, and 3111 cm raised to 1 in FTIR and Raman. The methyl group vibrations were expected about 3000 cm raised to -1 and the calculation and calculated as 3037 cm raised to -1 in CH and give CH3 stretching mode. In amines, the NH vibrations occur in the region 3500 to 3300 cm raised to -1. Here NH stretching is calculated as 3505 cm raised to -1. Also NH coupled with other vibrations were calculated as 1669 1628 cm per centimeter. Aromatic nitro compounds have strong absorptions due to the asymmetric and symmetric stretching vibrations of the NO2 group at 1570 to 1485 per centimeter and 1370 to 1320 per centimeter. Hence, the NO2 stretching vibrations of the molecule were calculated as 1339, 1322, 1333 per centimeter. The vibrational assignments were correlated well with the previous reported literature. FMO analysis, frontier molecular orbital analysis. The highest occupied molecular orbital or HOMO and the lowest unoccupied molecular orbital LUMO are known as FMOs, which is used to determine the way in which a molecule interacts with the other species. The FMOs of the molecule is shown in the figure and the FMO related molecular properties were calculated and shown in the table. The calculated low energy gap value 3.12 electron volt confirms that 6m8NQP molecule has higher chemical reactivity and explains the intramolecular charge transfer interaction within the molecule, which influences its biological activity. The energy needed to remove an electron from the filled orbital is known as ionization energy, and the energy released when an electron is added to an unfilled orbital, that is known as electron affinity, and that is equal to minus E LUMO. The calculated high ionization energy value 6.33 electron volt and low electron affinity value 3.21 electron volt of the molecule reveal the possibility of electrophilic and a nucleophilic attack respectively. The global hardness, global softness, electronic chemical potential, and global electrophilicity index of the molecule were also calculated. The calculated higher hardness 1.56 electron volt and lower softness 0.64 electron volt value indicate the stability of the molecule. The low chemical potential value minus 4.77 electron volt and the electrophilicity index value 7.29 electron volt of the molecule also confirm the chemical stability of the molecule, which is comparable with the potential bioactive molecule. UV visible spectral analysis. The study of UV visible spectra is a powerful method to obtain the characteristic information about the electronic states of the molecule. The calculated optical parameters such as excitation wavelength, excitation energy, oscillator strength, and the corresponding optical contribution of the electronic transition are listed in the table. The simulated UV visible spectra shows two peaks at 210 and 290 nanometer, and the simulated UV visible spectra shows main transition with maximum absorption weak for the peak 290 nanometer. This is due to the electronic transition from HOMO to LUMO, and its transition is assigned as N2 pi star transition of the title molecule. NBO analysis. Hydrogen bonding intra and intermolecular charge transfer interaction taking place in the molecule can be analyzed using NBO analysis. The higher stabilization energy value was obtained for the interaction between LP of N29 and sigma star O25 to O23 orbital, which confirms the intramolecular charge transfer ICT in the title molecule. That also the table shows the higher stabilization also mainly arises from lone pair of N28 to sigma star O22 to O24, LP of N30 to pi star C34 to C36, and LP of N33 to a sigma star O39 to N10 transition, which is responsible for the ICT of the title molecule. The stabilization interactions between LP orbitals and antibonding pi orbitals are common molecular feature for the pharmaceutical compounds. Hence, the results evidence that ICT in the molecule will lead the molecule as more reactive and bioactive. Mulliken atomic charge distribution analysis. The Mulliken atomic charge distribution of a molecule has significant influence in its dipole moment, polarizability, electronic structure, vibrational modes, model of electrostatic potential outside the molecular surface, and also describes the process of electronegativity equalization of the molecule. The calculated Mulliken atomic charge distribution values of the molecule are shown in the table. The Mulliken atomic charge distribution of the molecule shows that all hydrogen and carbon atoms have positive charge values. Further, the Mulliken atomic charge distribution analysis confirms that the hydrogen atom H11, H12, H13, and H14, and the electronegative atoms N10, O23, O27, O22, O24, O25, and N29 are more possible sites for the electrophilic and nucleophilic attack respectively. Molecular docking analysis. Molecular docking is an important tool used to identify the interaction between a small ligand and target protein, which has been widely used in pharmaceutical research for structure-based drug design. The lowest energy docked pores of protein ligand complex, including 6m8NQP P38 MAP and 6m8NQP top 2 alpha, were shown in the figure. The dotted yellow lines indicate the hydrogen bonds formed between the ligand and the targeted proteins. The corresponding hydrogen bond length values between ligand molecule and amino acid residues in the targeted protein are shown in the figure. The title molecule exhibits the lower binding energy value of minus 5.61 and minus 5.22 kilocalorie per mole for the targeted proteins M38A, MAP, and TOP2 alpha respectively. However, the inhibition constant value were calculated as 102.01 and 109.05 μm respectively. The obtained lower binding energy and inhibition constant value of 68N-QP-P38A MAP complex compared with that of TOP2 alpha evidence that the title molecule has potentially inhibit the P38A MAP protein. Hence, the present study will be useful for the development of novel drugs in the treatment of cervical cancer. Conclusion: the quantum chemical, vibrational, and molecular docking studies of 6M8-NQP molecule were performed. Vibrational spectra of the molecule were simulated, and the important vibrational modes were assigned. FMOS analysis shows that the title molecule has low energy gap value, which evidenced the ICT interaction from 6-methoxy-8-nitroculene to picric acid in the title molecule. The Mulliken atomic charge distribution analysis confirms the possible sites for the electrophilic and nucleophilic attack in 6M8-NQP molecule. The natural bond analysis also confirmed the ICT interaction 6-methoxy-8-nitroculene to picric acid, which leads the bioactivity of the molecule. The molecular docking studies conclude that the title molecule has a strong binding affinity towards the targeted protein P38A MAP associated with cervical cancer. Hence, the present study paves the way for development of novel cervical cancer drug. And these are some of my references. Thank you.
Okay, Geeta, any questions? Any questions? Any questions from the participants who are listening to the talk? Okay, Geeta. Geeta, can you hear me?
Yes, ma'am.
Yes. Okay, that's a nice, very good work.
Thank you.
And actually, this topic is very relevant. Drug for cervical cancer, medical a work of medical application, and you have identified this drug, knowing that it is what you have found in the result. Can you show the conclusion page?
Is it visible, ma'am?
No, it's not seen. Okay,
not visible.
No problem. I was asking that what you have concluded in the work. Were you aware that this sample is really having this property, and you were coming to the conclusion that you expected at the first time of the work itself?
Yes, ma'am.
You were expecting this result, or are there literature showing that this drug is actually having this molecular docking property that is binding?
No, ma'am. Actually, experimentally, I have concluded, and this is only my theoretical part.
Okay. You have done the theoretical part. Okay.
Yes.
So, Geeta, nice work. And
yes, ma'am.
A very good presentation. Wish you all the best.
Yes,
okay,
thank you, ma'am. Thank you.
We'll go to the last presentation. It's Amrita Krishnan B. She will be presenting her work on realizing photophysical response in Samaria SM3 plus activated Li3Y3T2 O12 phosphor for lighting and temperature sensing. Amrita Krishnan, are you there?
Yeah, ma'am.
Okay.
Am I audible, ma'am?
Yes. You can start the presentation.
Am I audible and the slides visible?
Yes, we can see your slides and also you are audible. You can continue.
Good morning, all. Okay, thanks. Good morning, all. Myself, Amda Krishnan B from Department of Physics, University of Kerala. Okay, first of all, I would like to thank the organizers of ICR THP for giving me an opportunity to present my work entitled as Realizing Photophysical Response in Samarium 3+ Activated Lithium Yttrium Tellurium Oxide Phosphor for Lighting and Temperature Sensing. Nowadays, there's a great demand for the emission tunable phosphors for light emitting diodes, LEDs, as well as ratiometric temperature sensors for non-contact thermometry. Regarding white LEDs, a red light is a critical component to obtain high color entry index, that is high CRA, and low correlated color temperature CCT. We know for the commercially available yellow phosphor like black cerium, its efficiency is decreased due to the lack of proper red. Lot of research is going on in this direction to incorporate a proper red component in order to increase the efficiency of the commercially available white LEDs. Antimony rare earth ions, samarium 3+ usually exhibit red emission due to its characteristic 4G 5822 transitions, which it can be from 5822 to 1152. So samarium 3+ based red phosphors can thereby increase the overall efficiency of the LEDs. And apart from the solid state lighting applications of phosphor materials, nowadays these class of phosphors are being used for the Hello, ma'am.
Amrita, can you present a little slower?
Yeah, okay.
I think you are too fast.
Okay, sorry, ma'am. I will try. So, apart from solid state lighting applications, nowadays the phosphor class can be used for the non-contact thermometry, particularly these optical temperature sensing purposes. And those class of phosphor materials that is being used in optical temperature sensing are termed as thermographic phosphors or TGPs. And the peculiarity of such thermographic phosphors is that it should have a temperature-dependent luminescence properties. And regarding this non-contact optical thermometry, they're very advantageous than that of the contact thermometry because it offers high detection sensitivity, spatial resolution, short acquisition time, better accuracy, signal discriminability, etc. And moreover, the samarium 3+ based phosphor materials are little studied for this optical temperature sensing. And among phosphor family, garnets a unique position owing to structural as well as compositional diversity. The general stoichiometry of the garnet structure is A3B2C3O12, where A, B, C corresponds to dodecahedral, octahedral, and tetrahedral site. So these all factors motivated us to choose the host material as lithium yttrium tellurium oxide, where lithium occupies tetrahedral, yttrium in dodecahedral A site, and tellurium in octahedral site. So, lithium yttrium tellurium oxide as the host material and samarium 3+ as the activator. So, samarium 3+ is introduced into the dodecahedral site, which is being occupied by the yttrium 3+. So, in this manner, my work is proceeding. So, the phosphor material was synthesized by solid state reaction route, where calcination was done at 850 degrees Celsius for 4 hours. The crystal structure as well as the phase purity of the samples were studied by X-ray diffraction, and the ritual refinement profile of the sample is shown here. From the pattern itself, it is clear that there is no secondary phase is decreased. So it is highly crystalline in nature. LVGA material is crystallized into the cubic Ia3 space group with lattice parameter 12.2 angstrom. And moreover, we can see that with the introduction of the submarine triplet, there is no point in the change in symmetry associated with this host material. So there is no problem, and we can proceed this with the phase pure nature of the samples. And further from the DRS spectra itself, it is clear that there's a strong absorption in the near UV region with a direct band gap of 4.6 electron volt, which corresponds to the fact that the present material is having a very strong absorption in the UV region, and thus it can be applied for the LED applications, solid state lighting applications. So we went for the photoluminescence analysis.
Hello, something has gone wrong. Veena?
Okay, am I audible? Okay, okay. Yeah, okay. Okay, ma'am. Shall I continue?
Yes, continue.
Hello, Anima ma'am.
Yes, Veena.
We think some network issue.
Something has gone. Oh.
Yeah,
Therefore,
actually I am discussing the photoluminescence analysis. I will start from the photoluminescence excitation as well as the emission spectra is shown here.
Amrita, can you hear me? Amrita?
Are my slides visible, ma'am?
Your slide is visible, but we are seeing the first page. The introduction page. Introduction, no, the title page.
Yeah, I can hear.
Ma'am?
Yes.
We think some network problem is there for her.
Yeah, where is she actually sitting? In the campus or at home? She is at the university campus?
Ma'am, actually, I'm Indra. I'm actually there's having some network issues here. We are working on to change it. She's actually she was left from the meeting. We are trying to join in.
Oh, okay.
Indija?
Ma'am, actually,
Hello,
Amrita is unable to unmute or share. Is there any settings change in the program?
No, you do one thing.
Yeah.
You get into the meeting through your mobile.
Okay, ma'am.
The audio will be shared through mobile and video will be shared through laptop.
Okay, ma'am. Okay.
Mute the laptop also.
Am I audible, ma'am, now?
First of all, you mute your laptop. Enter into the meeting through the mobile. Share the audio through mobile.
Okay, ma'am.
Am I audible now? Hello?
Yes, you are audible.
Okay. Okay. I hope slides are visible now and it is showing photoluminescence analysis.
Yes.
Okay. As we can see, the characteristic transitions of the samarium 3+ can be seen here, in which the maximum impedance band is obtained at a 611 nanometer of the samarium 3+, and also the other transitions 4G5/2 to 6H5/2 as well as 2/6H9/2 are also present. And also we can see that from the concentration dependent samarium 3+ emission spectra, with increase in the concentration of the samarium 3+, the emission intensity increases up to 0.05, beyond which its intensity decreases due to concentration cringing. And through Blaise expression, we have found out that this concentration cringing is due to the electric dipolar interaction that is taking place between samarium 3+ ions. And the critical distance for this is 20.72 angstrom. Moreover, we have further calculated the CA coordinate in order to understand where this emission falls. And from the CA coordinate of this optimum concentration sample, it falls at 0.45, 0.36, which corresponds to the overall orange red emission of the LYTA samarium 3+ 0.05 phosphor. And further, we have measured the internal quantum efficiency using integrating sphere, and it is found to be 35%. Thus, and also from the Mechanis relation, correlated color temperature is calculated as 2346 Kelvin. So these all results shows that the present material LYTA samarium 3+ is an efficient red phosphor which can be incorporated with the commercially available LEDs to enhance the color rendering index of such commercially available LEDs. And the interior transitions is shown in the energy level diagram as shown in the figure. So what we are dealing with is the solid state lighting part. Now we went for the temperature dependent decay time analysis in order to find out whether this particular material can be utilized for the sensor applications or not. So temperature dependent decay time analysis is carried out. As we can see from the figure that with increase in the temperature from 80 Kelvin to 500 Kelvin, there is a decrease in the decay time from 1.08 to 0.165 millisecond. That means here we have a temperature dependent decay time. As I have already told in the introduction part, those phosphor material that is having a temperature dependent photoluminescence properties are termed as thermographic phosphor. So this particular feature of the present material, which is having a temperature dependent decay time, temperature dependent reduction in the decay time meets the essential criteria for the sensor application. Thus, we have calculated the temperature, we have calculated the decay time at each temperature by fitting it with a bi-exponential function, and further using the Strack-and-Fogan model given by tau t equal to 1 by a plus b e raised to minus e by kt, where tau is the decay time at each temperature and they are constants. We have further fitted this with this model, and further we have calculated the sensitivities. And the relative temperature sensitivities in the range 200 to 500 Kelvin is plotted here. And we can see that with increasing the temperature from 200 Kelvin onwards, relative temperature sensitivity increases and up to and reaches the maximum at 0.5% per Kelvin at 370 Kelvin. And beyond which it decreases to 0.2 at 500 Kelvin. Thus, this particular material is showing a good temperature sensing properties in 200 to 500 Kelvin range, and thus we have made a comparison of temperature sensing properties with already reported materials so far. And as calculated here, we can see that the present material is showing good performance of the optical temperature sensing. So, in a nutshell, we have seen that present material lithium yttrium tellurium oxide samarium 3+ can be used as a potential red emitting phosphor to be incorporated in the LED application, as well as can be used as a sensor for the optical temperature sensing. So, concluding, lithium yttrium tellurium samarium 3+, which is exhibiting characteristic samarium 3+ emission, has an overall emission in the orange red region. Thus, it could be incorporated for the fabrication of LEDs with a better color rendering index. And to the temperature dependent decay time analysis, the ability of the LYTA samarium 3+ as a potential candidate for the optical temperature sensing is also investigated for the first time with a maximum sensitivity of 0.5% per Kelvin at 370 Kelvin. So, Francisco, we would like to fabricate a sensor based on this LYTA samarium 3+ material, as well as the thermochromic response of the present material as a candidate for the safety zone application is to be done. And I would like to acknowledge DST, Government of India, for providing me INSPA Fellowship, as well as State University of Kerala for instrumentation facility. And once again, thank you, IRCT HP 2021 Christopher College, Maranallur, for providing me an opportunity to present my work. Thank you all.
Okay, Amrita, your presentation was very good. Your work is very good. And participants, anyone have any doubt? Any questions? We were listening to potential material which can be used in the fabrication of LEDs in the orange red region. Any questions? Okay, Amrita, it was very good, and wish you all the best.
Okay, thank you.
Okay, so let me conclude the session very first, because I have encroached about 10 minutes of the next speaker. The session was very good. Hope all have enjoyed. We had a very good time and a very informative session. Okay, we'll conclude now. Okay, bye. Thank you for giving me this opportunity to the organizers, and it was a nice session. Okay, Veena, I'm stopping now. Veena, are you there?
Yes. Thank you, ma'am. Thank you.
Okay.
Presentations and providing valuable guidance to the presentators. Next, let me welcome Dr. Ganga Biji for the invited speech. Dr. Ganga Biji is currently a postdoctoral research fellowship at National Taiwan University. She completed her PhD.
Veena, I think your mic is mute.
Postdoctorate from the University of Johannesburg. Her research areas include nanostructures, optoelectronics, magnetic materials, and magnetism. She has several publications to her credit and is cited about 56 times by independent authors. Handing over the session to you.
Hello.
Yes, ma'am.
Okay. Good afternoon, everyone.
Good afternoon.
Okay. Myself, Ganga. Today I'm presenting mapping of electronic structure at the interface of semiconductors using cross-sectional scanning tunneling microscopy. So as an introduction, STM is basically STM scanning tunneling microscopy is a microscopic technique, and it is based on quantum tunneling. So quantum tunneling, as everyone knows, it's the tunneling of electrons through a potential barrier. And in 1981,
These 4 scientists at IBM's research laboratory combined vacuum tunneling with scanning capability and developed the first STM in the world. And they got Nobel Prize in 1986 for the development. And STM allows investigation of surfaces at atomic scale. So here we have a sample and tip that can measure the tunneling, that should be approached to measure the tunneling current. So the sample is basically a metal, and in metals, electrons are filled up to an energy level called Fermi energy. And when the tip is brought close to the sample surface, electron can tunnel through the barrier. Here, air will act as the barrier. And so when we apply an voltage across the tip and sample, we can measure the tunneling current. And this is the graphic image of STM. And there are many components. So we should have a sharp and clean tip to measure the tunneling current and the sample. And the tip is attached to the piezoelectric tube. And there are many electronics to control this one. And this distance control and scanning unit control the distance between tip and sample and measure the tunneling current. And there is a data processing unit which processes the tunneling current and display the data. And the tip, a sharp tip is very desirable for imaging the surface of the sample. Mostly the tip is made up of tungsten metal or platinum iridium alloy. And this is the SEM image of a tip at 100 micrometer resolution and 200 nanometer. Basically, should be the tip should be 1 atom thick, so that we can measure the exact atomic resolution. And the tip is attached to a piezoelectric scanner. So piezoelectric effect, when we apply an electric field, it can change the shape of the material. So by applying a voltage, we can move the tip along XY direction and Z direction. So in that way, we can control the movement of the tip along the sample surface and the height between the sample and the tip. So the piezoelectric scanner basically control the position of the tip along the sample surface. And there is distance control and scanning unit. This place the tip close enough to the sample surface before the scanning measurements start. And there is data processing unit. This record the tunneling current and control the voltage to the piezoelectric tube and produce a three-dimensional map of the surface. And a vibration isolation system is also required, which since the tunneling current is very sensitive to the sample's tip distance. So in order to reduce the effect of inner and outer vibrations on the tunneling current, it should be isolated from the outer environment. Coming to the working principle of STM, first we apply a bias voltage to the tip, and the tip is brought to the sample surface. It should be within 4 to 10 angstrom range. So the sample tip distance is basically 5 to 10 angstrom. So then because of the bias voltage, the tunneling current start to flow, and it is measured and amplified by the amplifier. And there is a feedback loop included in the system, which can adjust the height of the distance between sample and tip based on the measured tunneling current. So we preset a tunneling current value. So if the preset value, if the measured tunneling current value exceeds the preset value, the tip sample distance can be adjusted accordingly so that the current goes to the preset value. In that way, we can get an information about the sample surface. So by moving the tip across the sample in both XY plane, we can get a three-dimensional image of the surface. And there are basically there are two modes of operation in STM. The one is constant current mode and other one is constant height mode. So in constant current mode, the tunneling current is keep constant and the tip position is varies accordingly. So we get a topology of the surface according to the variation of the tip. And this is the most commonly used mode in STM. And another one is constant height mode. In this mode, the tip position is held constant and the tunneling current is measured accordingly. So this gives the information about the surface. So in constant height mode, a flat surface is required. Otherwise, there is a possibility that the tip can crash. So in the case of rough surfaces, mainly we focus on constant current mode. So STM basically does not give the exact image of the surface as we observed in optical microscopy. So it measures the local density of state at the surface. So this is the atomically resolved STM image of silicon 111 surface. So this is the unit cell and you can see the atomic resolution in this image. And this is measured at 0.5 volt bias voltage. And other than microscopy, there is spectroscopy also. In microscopy, we get information about the topology or surface, information about the surface. While in the case of spectroscopy, we get information about the electronic structure. So in this case, the tip is positioned to a region where we need to get the electronic structure, and the distance between the tip and the sample is fixed. In this case, we vary the bias voltage and measure the tunneling current and as a function of bias voltage. So at low bias voltage rate, we take the differential conductivity, the differential of tunneling current versus the applied bias versus applied voltage is proportional to density of state. So we get information of density of state of the surface using scanning tunneling spectroscopy. So this gives information about the band gap, conduction and valence band. So this figure shows how the STS image will be. So this is basically a semiconductor material and there is Fermi level, in-gap state and conduction and valence band. So when we ramp the bias voltage, we can measure the density of states like this. So it gives information about the band gap, in-gap state and conduction and valence band. And this figure shows the tunneling spectra of N-type gallium arsenide from both experiment and theory. So you can see the experiment is this blue curve and it matches exactly with the theoretically simulated values. So this gives the accuracy of the STM measurement. And so far we have been talking about measuring the tunneling or topology of the sample surface. But in the case of when two dissimilar metal or semiconductors are combined, if we need to study the electronic structure at the interface, then we need to have another technology. So this is called cross-sectional tunneling microscopy. So in this case, if we have a substrate and film, we just cleave the sample like this, like break the sample, and the STM measurement is performed at the interface. So we get information about the electronic structure at the interface with atomic resolution. For this, we need a well-ordered, clean and flat surface. And usually the sample is cleaved in ultra-high vacuum and the measurement is carried out at the interface. So this is an example of how the band or electronic structure is mapped at the interface of two dissimilar metal. So this is a complex oxide interface, LaAlO3 and SrTiO3. And neodymium STO is a substrate, and it acts is the substrate, and there is a STO and LAO. LAO is only 5 unit cell, and it has been reported that even though STO and LAO are wide bandgap semiconductors, the interface can produce highly conductive interface can be produced. And SRO is another thin layer coating, just 2 nanometer to make the surface conducting. And this is the DIDV map. So DIDV map is obtained by obtaining the DIDV curve at different region of the sample surface. And so we can see a contract difference in this region. So this enhanced, so here it indicates that there is an enhanced charge at this region. So this is the accumulation of charge carrier at the interface between STO and LAO. And this is the STM image. And here also there is a contrast difference. It shows this charge accumulation is only for a region. It's called a decay length lambda. And so this shows the STS scanning tunneling spectroscopy curve measured at the different region of the sample. So this color defines the STS spectra measured from different region of this this sample. And so the violet curve represents STS measured far away from the interface, and blue near the interface. And the same way, the red curve represents the spectra measured at the interface of STO, and the brown curve represents the spectra measured far away from the interface in the STO region. So from this, they have calculated the position of unoccupied and occupied state, and it is for STO. They have taken the red and brown color curves, and it is calculated to be 1.7 and 1.45. So there is a change in the position of occupied and unoccupied state for STO near and far away from the interface. So at the interface, there is a reduction in the decrease in the position of occupied and unoccupied state. So similarly for LAO also, the position of unoccupied and occupied states are measured, and it's also estimated to be 2.3 and 2.9 volt. So there is also a change in the position of this state when we move from the interface to far away from the interface. And this is the information combined from the previous slide. So this is the distance across the sample, and this is a sample bias. So this is the 0 is the interface between STO and LAO. And so here we can see, so the position of the band occupied and unoccupied states are measured to be 1.7 and plus 1.7 and minus 1.7, and there is a decrease in the position of this state while approaching the interface of LAO. So this change is approximately estimated to be 0.8 nanometer, and this represents the band gap state penetrating to the metallic surface. And similarly, in the case of LAO, there is an increase. There is also a change in the position of this state at the interface, and this represents, and from the difference in the position of this state, the built-in electric field can be estimated, and it is calculated to be 30 milli electron volt per angstrom. So in this way, we can calculate the band position and the built-in electric field at the interface of two dissimilar metal. Here I am showing only the interface of two dissimilar metals. So to conclude, a cross-sectional STM provide atomic resolution investigation of the electronic properties at the interface. So in the recent days, band engineering is an essential tool for obtaining novel properties in material. So in that case, STM provide atomic resolution electronic structure, which can provide understanding of the structure when we artificially define heterostructure. So this can be very effective in band engineering in advanced methods. So thank you.
Thank you, ma'am, for your valuable insights. It was truly an enriching session that made us look deeply into the topic. The session is now open for questionnaire. You can either raise the hand or unmute yourself and ask your questions. You can also put your questions in the chat box.
Ma'am, there was a question in the chat box. Can I read it?
Yes.
Ma'am, could you please describe your journey to be a postdoctoral research fellow at NTU?
Okay. So I completed my PhD from IIT.
Please unmute.
Hello, sorry.
Yeah, you are audible now.
Oh, okay. So I completed my PhD from IIT Madras. So where I was working on magnetism of nanostructures. So after that, I joined in the University of Johannesburg, where I was studying the the electronic structure of two-dimensional materials. Then after that, I joined National Taiwan University, where I am studying this the surface of two-dimensional material using scanning, tunneling, microscopy, and spectroscopic technique.
Any question undo?
Actually, this is not my project. I'm working on superconductors. So study the superconductivity using scanning, tunneling, microscopy, spectroscopy technique. So I just choose these two present. So it's not my topic of research.
Ma'am, one last question.
Okay.
Ma'am, if you could point out a person who motivated you to achieve this much at such a young age, who would it be?
Of course, my PhD supervisor, Dr. P.N. Santosh. So he has been very helpful and guiding me while I was a PhD student at IIT Madras. So,
thank you, ma'am.
Okay. Thank you very much.
The questionnaire session has come to an end. Thank you, ma'am, for your valuable insights once again. Now it's time for lunch break. We'll continue this conference by 2 p.m. The participants should make sure to join the session again by 1:50 p.m. Thank you.